Very good day to everybody. If you are watching this video, I'm going to demonstrate how our drug discovery module is going to work. It's something kind of innovative for the biomedical research and the synthesis part, and recreating things actually. So actually what it does, it's a typically an intelligent literature which also having the mining engine to take or rather process various numbers of or rather the thousands of papers. Okay, so how it's actually going to work? So like for instance, we have created a C2 drug discovery file. So it's present over here, and these are the file utils that we are normally using. So for instance, few things, it's some of the technical parts. We are using 4 different things. Okay, and that's one is a data loader. We are pushing the datas. It's all written in Python, and yeah, data loader is currently under building, so this sections will be taken care of later on. But this is the most important thing, which is the image processor. I'll going to get back into this part a few minutes back. So yeah, this is our PubMed feature. Okay, now what we are doing is that we are requesting few of the site utils from the PubMed research NCBI research center, as we have got the research key officially. So we can take the research data and we can push into it to recline or rather refine our things, right? So yeah, and it's a technical, what we are actually returning the values, that is one article ID, second title, and third is the abstract. So this is the main important, because these threes are the most important thing that actually gets processed for the NLP, rather the natural language processing jo model is there. So that's the main important thing. Okay, fourth comes the text processor. How does it analyzes? So we are using one model. It's a very lightweight model for now being. We are using a Spacey, or rather it's in the Hugging Face site, so of course it's a very good site. And Spacey is also used for both, you can say fast and or rather the prototyping kind of thing of natural language processing models. You can also create, LLMs are there, so you can use this Spacey module to push through it, right? So yeah, we are using that to find all those important, as we are actually taking 3 main important thing. We are not pushing for right now, that is diabetes disease pattern. So yeah, and you can see over this things, I believe, yeah, disease patterns. So one, we are registering cancer, second is the tumor, and third is the neoplasm. So biological people might know, I'm sorry that I don't know this, but yeah, I'm the technical part, so yeah, it does work. And we have also pushed a lot of things over here. For example, for the bone thing, we have pushed arthritis, neuropathy, and for mental or rather the psychological part, hypertension and hyperglycemia, and yeah, and lymphoma and glioma is for the pathological part, right? So rather the blood part. So yeah, we are pushing this limit. Yeah, we made it, but it's kind of prototyping. It's a V1 project, as because like if you properly see over here in the drug discovery section, so yeah, it's a version 1 tool. Rather this is a version 1 tool that I am actually building, rather the compressed it. So yeah, it's kind of like that. Sorry. So yeah, so this is the thing, data loader is going to be uploaded. This is a synthesis scorer. Now how does the synthesis scorer works? It actually works on the relevance, novelty, and methodology, and it actually synthesize the research paper into which one should be the best, or rather the citations, or rather the public audience captivations are so much into it, you understand, right? So these 3 portions are gonna get covered and will rate how is it the papers are. Okay, so yeah, and this is the path model. It's a very important model. Now I should explain why we are using path model, right? So path model actually has, we are using GPU. Till now we are being capable to the down part. We are using the RTX machines, RTX 2050, which is having certain 1800 CUDA cores, and we need to run this in a very high GPU end. So I probably computational scores must be there to run a proper to get a proper feedback from the things, right? So yeah, um let me show you how does let me show you one thing that I have kept it aside. This is a surgical pathological image that I have pushed in the raw file. You can see this. So it actually helps to recreate this thing and to search this. What we are using is, let me show you one. We are using a public synthesized and a refined model, that is the Hugging Face Google Pathological Foundation. It's a free to use, not probably free to use totally, as because few things are not to be disclosed. Yeah, so we have got the API key over here. We have pushed in the API key section over here in the Path Foundation API.txt. I don't want to show it this for the security reasons, but yeah, it's kind of like that. So this pathological file, we are pushing it. So I have put the main file over here, main.py. What we used to do is that we have to run this, right? So this is a sample pathological JPG. So it will help to find the features and the mean features, okay, in the decimal and the integer values. Okay, so let me push it. Hold up a sec. So Python main.py. So let me show you in a bigger screen, right? It might be visible, right? I'll let it process. It will take some time for the GPU to work. So these are the current limitations that we are facing due to lack of GPUs. It might work. Yeah, the PyTorch has started completely. So we are now threshing hold to the 50 articles. We can go till thousands. We can do thousands of articles. And our medical NLP model tags chemical structures, okay, with some biological targets with 92% accuracy. So the citation work is completely done, and the theoresis is done over here. See, you can get the score over here probably, right? So for instance, I have searched for cancer drug discovery. Now it's not typically working, as because for the drugs it's not totally 100% accuracy, as because the file size is only 5.5 5 GB. Let me show you. Hold up a sec. So this one, right? Sorry, local disk H, sorry, yeah, this one. So we are currently pushing. It's a RAR file that I compressed. So normally it's a 5 GB file. If you can see 5.38, and 5.38 is never accurate. It's having certain million parameters. You have to get at least 7 billion parameters. Even it's not totally 100% accurate. It's approximately 50 or 40% accurate. We need 180 billion parameters, just like the Olama models that we get. So just like that. So yeah, it's kind of working, as because the discovery of secondary metabolites for the sponge. Hold up a sec. Yeah, so it's kind of working like this. You see, diseases can be get hold into it. Cancer, it searches for those terms. So yeah, it does work. And for the image analysis and the feature shape, it shows 1,768 and mean features as -0,02. So So it's kind of working like that. So let's refine it a long bit. Hope it does works, and I believe it will work. Normally there was a output that I predicted was as this was the only one file that I've got as a lead compound with this type of risk managing and the synthesis pathway. So it's kind of good. And what we have used over here is we have used PyTorch for deep learning models, Spacey for the medical text processing, Hugging Face transform for molecular generation. Till now we have not implemented that, and RDKit for chemical validation. Till we have not implemented that. So yeah, we have to use as because you can see over the requirements file over here. So yeah, these are the modules that we have used and we are going to use in a future time. So we are just scratching the surface. Next phase must integrate. Hope so. So thanks and have a very nice day ahead.